Welcome to the deep dive. Today, we're looking into something pretty exciting in green chemistry.
Yeah, it's about a new way, potentially much greener, to get useful stuff from lignin waste.
Lignin waste. Okay, I know lignin's in plants, makes them strong, right? But break it down for me.
Exactly. It's basically the structural glue in plants, particularly wood. Super abundant. But here's the kicker.
Uh-huh.
We throw away almost all of it. Something like 98% of lignin from forestry operations.
98%. Wow.
Yeah, it adds up. About 100 million tons every year, just from forestry and agriculture combined. Gone.
Okay, that's staggering. So what's the mission for this deep dive then? What are we trying to figure out?
Well, we want to unpack this new discovery in enzyme
Hmm?
and, understand why it's such a potentially big deal.
An enzyme.
Yep. One that can unlock valuable molecules hidden inside all that discarded lignin.
And these molecules,
Yeah.
what can they be used for?
Oh, all sorts of things, like fragrances, flavorings,
Uh-huh.
even potential biofuels or building blocks for pharmaceuticals, basically turning waste into resources.
Right, transforming waste, which definitely sounds better than how we usually get these kinds of chemicals,
It really does. Current methods often rely heavily on petroleum.
which is non-renewable, obviously.
Exactly. And they often use heavy metal catalysts, which, can be toxic and environmentally problematic.
So this new approach offers a cleaner path.
That's the promise. It uses this newly found enzyme and hydrogen peroxide.
Hydrogen peroxide, like the stuff you get at the pharmacy.
Sort of, yeah. It's a relatively simple and much more environmentally friendly chemical compared to the traditional heavy stuff.
Yeah.
It helps power the enzyme's reaction.
Interesting. So where did this wonder enzyme actually come from?
Scientists found it in a soil bacterium. It's called Imicolatopsis thermoflava.
Imicolatopsis thermoflava. Catchy.
Yeah.
And what makes the enzyme it produces so special?
Well, the key thing is how it deals with lignin, especially hardwood lignin. Hardwood lignin makes up roughly half of all this waste material. And it has these complex molecular structures. Previous biological methods, other enzymes, they could really only break down one type, the kind also found in softer woods.
Ah, so they had a wall with the hardwood stuff.
Pretty much.
Yeah.
This new enzyme, though, it's the first biological tool we've found that can efficiently tackle that second, more complex component unique to hardwoods.
So it's like it unlocks a whole new part of the lignin molecule that was inaccessible before, biologically, I mean.
Precisely. It can process this significant chunk of lignin waste, maybe 50% of it, that was previously resistant to these gentler biological methods.
That does sound like a breakthrough. Not just another enzyme, but one that opens up a huge feedstock.
Exactly. And it's seen as more than just this one specific reaction. It's kind of a model
A model for what?
for developing future enzyme factories or biorefineries, using biological catalysts, like this enzyme, to do green chemistry on a larger scale, taking lignin, maybe other plant waste too, and making valuable chemicals cleanly.
Right, avoiding those harsh traditional methods you mentioned.
Yeah, the current industrial ways to try and get value from lignin often involve, well, pretty brutal conditions.
Like what?
Really high temperatures, very high pressure, sometimes strong acids, or even poisonous solvents.
Oof. Sounds expensive and not great for the environment.
It is both expensive and environmentally challenging. So this biological approach, using an enzyme and hydrogen peroxide at potentially milder conditions, is a massive improvement.
Okay, so let's recap the core finding. We have this enzyme from the soil bacterium Echinopsis thermoflava,
Right.
and its superpower is breaking down a tough component of hardwood lignin, which is about half the waste,
Yep, using hydrogen peroxide.
offering a much greener, potentially cheaper way to get valuable chemicals compared to traditional petroleum-based high-energy or toxic methods.
That sums it up perfectly. It's a blueprint for a more sustainable chemical industry, leveraging biology to turn waste streams into value streams.
It really shifts the perspective, doesn't it, from waste disposal to resource creation.
Absolutely. Instead of just burning lignin or letting it pile up, we could be systematically converting it using these tailored enzymes. It taps into the idea of a circular economy.
It definitely makes you think. Lignin is everywhere, one of the most abundant polymers on the planet.
It really is.
So here's something to ponder. If we can find an enzyme key to unlock lignin's potential like this, what other biological materials, things we currently just discard or treat as low-value waste, might be waiting for their own enzymatic breakthrough to become sustainable resources? Definitely food for thought.
A great question to end on. The potential is vast.
Thanks for exploring this deep dive with us today.